the lab we're starting today is going to run all weekend. And what we are going to do is we want to prove that the force that carries water up the xylem of a plant originates in the leaves. Okay? That it is in fact an osmotic pulling force. Okay? That is the result of water evaporating, leaving salt behind, and creating that osmotic pulling action that will draw the water up. The way we're going to prove that is we're going to cut the leaves off the plants. And we're just going to stick the leaves and a bit of stem in some dyed water. Hopefully, over the course of the weekend, okay, we will see, well, we won't see it because we won't be here, but on Monday we'll see the evidence of it, that water will move into the leaf. Okay? The evidence of that will be that there will be dye in the leaf. Okay? The veins should be colored, and if you're really lucky, you'll actually see little dots on the bottom of the leaf indicating where all the stomates are. Okay? That often happens. Okay, so what we're doing is we are altering the plant, basically. Okay? We're cutting off the roots and only leaving a bit of stem and leaf, okay, in order to see if, in fact, the pulling force originates in the leaf. Okay? What we're observing is the appearance of the leaf, and we're also going to record the volume of water before and after. Okay? Those will be our two indicators of whether or not the plant was able to transport water. You guys are getting better at this. I see you all, I hear you all typing, and see you all typing. You're getting it, but I'm kind of going over the design right now. That's good. Okay. The rest of the experiment is that those plants will sit in that water, okay, and they will sit in that windowsill. Okay, so I want you to think about some things that are going to stay the same for that plant throughout the weekend. Okay, if it sits in that windowsill. Keep in mind, the heat stays on here over the weekend. They don't turn it off on the weekends to save energy. Okay, our hypothesis. Okay, so I've explained this twice already this morning, but the if part needs to talk about how plants transport water. Okay, what is the force that pulls the water up the plant? And you can include the polar nature of water part in that as well. Okay, but what we're really focused on here is where does the force that actually pulls upwards come from? The and part, as always, is going to describe the experiment where we cut some leaves and a section of stem off of a plant, place it in dyed water, and record the starting and ending volumes and the appearances of the plant. So we have some qualitative observations and some quantitative observations that we're making. Okay? The quantitative being the initial and final volume of water, the qualitative being the appearance of the leaf. So you're going to take some before pictures today and some after pictures on Monday. The then part, what's your proof? What's the evidence that that is going to happen? Okay, so you're predicting your results here. Then, on Monday, okay, tell me what's going to happen. Okay, so the procedure for today is going to work like this. Our end product is this. Okay? We're going to have a couple of leaves and a section of stem in a graduated cylinder filled almost to the top with dyed water. Okay? That's our end product. Right? There's a bit of steps that obviously go to that. It's not just going to appear. Okay? We have to build it. If we build it, water will be transported. Okay? So that's what we're looking for. Now, a couple of things to note when you're doing this. Look how far into the graduated cylinder the stem goes. Okay? It's really important that you cut off enough stem to reach down into the graduated cylinder. If you do not, enough water could be transported that the bottom of the stem would no longer be in the water by Monday. Okay? So we need to make sure that the stem goes fairly deep into the water so that that won't happen. I don't know if you can really see it very well, but there's something over top here of the graduated cylinder, and the leaf is poking out of it. Okay? So I'm going to talk about how we're going to put that on there and what it is and all of that here in a bit. You can tell that when we did this one, the weather was a lot worse. Okay, so the first thing you're going to do is make your dyed water. You're going to need 2 graduated cylinders. Okay? You're going to need a small graduated cylinder, okay? Probably a 25 milliliter one. Okay? And you're going to need a large graduated cylinder, probably 100 milliliters. Okay? So we'll get a small one and a big one. The big one has to be 100 milliliters. In that graduated cylinder, you are going to place tap water. Okay? Just tap water, fill it until it is at 100 milliliters. Okay? Now, how do you read that on a graduated cylinder? Okay? So on a graduated cylinder, okay, there are markings, okay, all the way up the side, and when you put water in it, because water is a polar molecule, it will cling to the sides of the graduated cylinder, making the top of it look a bit like that. We call that a meniscus. Where do you need to read your volume from? The top of the meniscus or the bottom? Always from the bottom. So you're going to have to get down right beside it, eye level, okay, and make sure the bottom of the meniscus is at or just below 100 milliliters. At that point, you are going to add food coloring to the water. Okay? Now, I used to, back when I was younger and crueler, put out all choices of colors. Unfortunately, people would pick green and yellow, which initially was funny. And then it was tragic, because they came to me on Monday and they're like, "Mr. Coderre, we can't see the food coloring." And I'm like, yeah, you're right. Plants are great. So I don't do that anymore. Okay? I just put out red and blue. So there won't be any of that going on. Okay? You can choose to just use red, or just use blue. You can be wild and crazy, and you can mix them up and get purple. Okay? Totally up to you. Okay? I would not use more than, let's say, like 6 drops. Okay? If you've got 6 drops in there, it's going to start getting kind of like thick looking. Okay? We don't need it to be like opaque. Okay? It needs to just be colored. So between 4 and 6 drops ought to do it. Okay? If it's still looking a little bit thin, you can always put in another drop. Okay? So that's what you're going to mix up. Then you're going to pour from the 100 milliliter cylinder into the smaller graduated cylinder. Now the reason we're doing this, because probably a lot of you are going, "Gee, that seems like double the work. Why didn't we just pour and mix everything in the small cylinder?" Well, the problem with the small cylinders and all graduated cylinders is the markings don't go up as far as I drew them here. On most graduated cylinders, the markings only go up about this far. And we need it to be filled higher than that. So there's no way for us to measure the volume of water with the smaller cylinder because we're gonna fill past the markings. Okay? So what I want you guys to do is fill that smaller cylinder until it's close to the top, but not all the way to the top. Why? What'll happen when you put the plant in? It'll spill over. And will you know how much spilled over? No. And then your measurements will be all messed up. So don't fill it all the way to the top. Give it maybe 2 centimeters, okay, or so from the top. That should be enough. And when you're putting your plant in, if you're seeing that it's looking like it's going to displace too much, then you might have to take the plant out and pour a little bit back into the original cylinder. Okay? So once you've filled the second cylinder, you need to record how much is left in your main graduated cylinder, in the large one, okay? Because that's going to tell you how much is in that cylinder, right? So you're going to go 100 minus whatever is left. That will be your initial volume. Okay, everyone with me there? So 100 minus whatever's in, okay, whatever is left in here, okay, that's your initial volume. And then on Monday, we'll just pour what's left back in the big cylinder and measure it, and we'll have our final volume. We won't even have to do any subtracting. We'll just measure it directly. Okay. Everyone all right with that? Okay, so if I just put a plant into, if I just like cut a piece off and walk over to my station and stick it in the water, what are some problems I might encounter? Yep, exactly. If I don't like create some kind of seal against the top of that graduated cylinder, water will just evaporate out of the cylinder, and I'll have no idea how much of it went through the plant and how much of it just straight evaporated out of the cylinder. So, to counteract that, we're going to create as tight a seal as we can over the top of the graduated cylinder. To do that, you're going to come up to the front, and I'm going to give you the finger off of a rubber glove. Okay? And you're going to put that piece of finger, rubber glove, over top of the graduated cylinder and poke a hole in it with your pen or pencil. Just a small hole. We want to be able to push the plant through that hole and have the rubber glove create a decent seal around it. It'll be way better than just having it open. It's not going to be perfect, but it'll be way better than just having it open. So that's how we'll counteract that part. If I just cut the plant off and carry it over, what can happen to the water in the xylem? It could drip out, but even more importantly than that, when I cut it, air could get in. And if air gets in, it will disrupt that whole polar nature of water adhesion, cohesion thing that the water's got going on inside of there, and we don't want that. Right? So what you need to do when you're cutting, there's a technique to the cutting. Okay? And the technique is, I have a sink full of water right now, and the plants are in it. You're going to hold them underwater. Don't worry, they won't drown. Okay? Just hold them under, okay, and cut off the part you want. Put your thumb over the cut end and go back to your station, and then really quickly take your time off, put it in. Okay? So you have to have the whole thing built before you go get the plant. You don't want your partner standing there with the thumb over the end of the plant, okay, for like 5 minutes while you get things ready. Have everything ready. The plant's the last part. Okay? Everybody pretty clear on this? I mean, it's not rocket science, but we got to be careful about how we do it. Okay? Think about all the possible sources of air that are coming in here while we're doing this. Okay? But that's what we're going to do. Okay? So there's going to be a few pairs of scissors. One person from your group can come up, and I'll help them decide how much leaf to take. Okay? And put in the graduated cylinder. All right? Okay. When you've built your device, okay, I'm going to come around with some masking tape and some markers. You're going to put the masking tape around the bottom of the glove. Okay, this one doesn't have that, but you're going to put masking tape around the bottom of the glove and write your names on the masking tape so you know which plant is yours on Monday, because on Monday there's going to be a lot of plants sitting at my windowsill. Okay, once that's done, you just bring that plant back here, put it on the windowsill. Oh, before that, sorry, you're going to take pictures of it. Okay, take a picture like this of it, and then take some close-ups of the leaves. so that you can compare what it looks like today with what it's gonna look like on Monday. All right, everybody with me there? So volume and pictures, that's what you need.